Hi, my name is Rita. I'm a physics engineering student and I'm here to explain to you in what my master thesis consists in. The title of my thesis is the development of a new PET detector module with improved depth of interaction and time of flight capabilities, and is a fusion between physics and medicine. This work is an example of how physics concepts can be applied in other fields. The merge, in this particular case, results in the development of new techniques which are crucial to improving healthcare. Better detection, better treatments. But first, what is a PET image? PET is short for positron emission tomography. It is a medical exam that allows us to obtain images of some mechanisms that occur inside our body. In other types of medical imaging exams, like an X-ray or a computed tomography, we have images that in a very basic way can be seen as photographs of anatomical structures like bones or even organs. In PET, we don't actually see these structures, but instead we can see if they are working in a normal or abnormal way by the amount of glucose that is consumed. If in some way you find yourself a little bit lost, think in glucose like food for our cells. Cancer cells need an abnormal amount of glucose in order to be always reproducing themselves, and we can use this behavior to detect them. In a PET exam, these regions will appear as bright spots in opposition to the black that represents lack of absorption. The PET exam starts with a contrast injection in the patient right before starting the image acquisition. This contrast has two very important characteristics. First, it has the same structure as the glucose, so that we can trick our bodies into absorbing the contrast in the same way. Secondly, it must be a radioactive contrast. That is, it is unstable and it will search for its stability by radioactive decay. The emitted particle in the radioactive decay is a positron that will locally interact with a neighboring electron. The technical name for this process is positron-electron annihilation, and the result is the emission of two photons traveling in opposite directions. This way, the pair of photons will reach two opposite detectors from the ring that is surrounding the patient during the exam. If we connect the two detectors by one virtual line, we can ensure that this line will cross the emission point that corresponds to the location of the tumor. However, without any other information, every point of this virtual line can be the origin of this photon emission. In order to obtain more information and narrow down the possible points for the tumor location, two techniques will be used. The first is time of flight. If we consider an emission point that is displaced from the ring center, we can easily see that the path traveled by each photon isn't the same. With this technique, it is possible to know from the same pair of photons if one of them arrives first to the respective detector. If that happens to be the case, we can also learn which one of them reached it first. Knowing their velocity, we can limit their emission point from the whole virtual line that we had in the beginning to an interval much smaller. However, we still have a component that is contributing to this uncertainty. An efficient detection of an emitted photon results from the combination of a scintillator and the photodetector. The photodetector that will detect the emitted signal is coupled to the scintillator through which the photon will travel and in which will eventually interact. Thus, the path that each photon travels inside the scintillator until interacts will condition the signal collected. And due to this is a very important information. The study of this component is the depth of interaction. The lack of these two informations contribute to poor image resolution and can ultimately provide wrong information about the tumor, mainly about its size. So the main objective of my thesis is to obtain a timing correction in order to eliminate these errors. At the end of the day, we want to locate the tumor with the highest accuracy possible. If these two types of information can be extracted with good resolution, the tumor region can be identified with a precision in the order of few millimeters, implying great advances in the detection and staging of these diseases.